what I'm going to move on to speak about now is the review questions that we got for the review questions that we had practiced that I put in Blackboard. So I'm going to talk about some of the good responses. So this is one of the ideal responses, and I'm not trying to put anyone on blast or shame anyone here. I'm going to point out some of the good responses. I'm going to point out some of the responses that were not ideal, okay, in an effort to point out what you should be focusing on to get full points. So this was a really good response. The first question asked about the barriers for reabsorption and secretion. And then it said specifically, be sure to include all plasma membranes that must be crossed for substances that are being reabsorbed or secreted. And so this was pretty straightforward. There wasn't a whole lot of detail required here. Simply the barriers are the apical membrane of the tubular epithelial cell, the basement membrane of that cell, the peritubular space, and then the capillary endothelial cell. Okay, so those are all the membrane or all the structures that must be encountered to be reabsorbed as well as to be secreted in the opposite direction. Some of the variability came about with the second question. So this was a pretty good response. Most of the absorption occurs within the proximal convoluted tubule. This occurs because sodium is transported out of the tubular space. So that's the first part of water reabsorption. Okay, the fact that solutes, specifically sodium, is being transported across the membrane first. And that happens passively according to the sodium-potassium pump. And then they went on to describe how chloride is moved across the membrane. So chloride is moved passively, and then sodium is pumped across the membrane, across the basement membrane. And then that sets up a gradient where water can move. Okay, so this is basically the important parts of this response. Talking about how sodium is moved passively into the cell, pumped actively across the basement membrane or the basolateral membrane. Chloride is moved actively into the cell, and it goes passively across the basolateral membrane. And then because of the movement of those two solutes, water moves according to its gradient or according to osmosis. And then the third part of the question, define the terms transport maximum and renal threshold. So the transport maximum is the rate of movement of glucose at which maximum reabsorption has been reached because the proteins that transport glucose have become saturated. Okay, so perfect response there. We've got maximum reabsorption, the transporters have been saturated, and it's really the rate of movement that we're defining here in terms of the transport maximum. Renal threshold is the plasma concentration of glucose at which glucose is not fully reabsorbed and starts to become present in the urine. So again, this is correct. So we're speaking here about the plasma concentration of the substance in question. So we're looking at glucose at which we no longer have full reabsorption, but we start to spill this solute into the urine, okay? So that was pretty straightforward, and that was a good answer. This was also a good response. This was, again, pretty straightforward. I won't go through the details here, but you just listed out the structures. And here I really want to point out that we don't have to go into a lot of detail. This person just listed out these structures, and that is fine. That's going to be okay in terms of responding there. The second part of this, again, was pretty short, but very succinct. So they went directly to the point. Chloride is actively transported, sodium is passively transported, then they are moved across the basolateral membrane actively and passively. They talked about this respectively, and that creates a gradient for water movement. And then the final part of the question was also very short, very to the point, very succinct. Okay, this response was okay, right? There were some aspects that were not, it was not enough detail. So for question 2, the general mechanism for water reabsorption is a passive process. It follows solutes such as sodium and chloride into the tubular cells and interstitial space. Water freely flows according to its passive gradient, allowing it to move freely into the capillaries. So this response needed a little bit more clarification, a little bit more detail as in terms of where the solutes moved, how they moved, the fact that they were moved actively across the basolateral surface in the case of sodium, and then passively in the case of chloride, and just a little bit more detail in terms of how these solutes were moved to begin with. And then we can kind of talk about how water follows. So this was an okay response, but a little bit more detail here would have been ideal. And then this last one definitely needed a little bit more detail. So this answer was water follows salt through osmosis on the proximal tubule, causing water to be absorbed. So it's not an incorrect response, but it's very vague, and this is not going to get you full points, okay? You wanna talk about the solutes that are moving, how they're moving, either actively, passively, and then how water is following. You also wanna talk about what's going from where to where, okay? Whether it's going from the tubular lumen into the cell, into the capillary endothelial cell, and then into the plasma. Again, so some more detail is definitely required here. And then, okay, so that was it for question 1, all right? I'll see if there are any questions that come up so far. Okay, I don't see any questions, so I'm gonna keep moving. Question 2 had 3 questions as well. So this is an ideal response. The first part of the question, briefly explain how an increase in the secretion of renin stimulates the secretion of aldosterone. How does an increase in aldosterone secretion affect sodium reabsorption? And so this response was pretty succinct. An increase in renin secretion stimulates aldosterone secretion because renin converts angiotensinogen inactivated into angiotensin 1 activated, which is then converted into angiotensin 2 by the ACE, right? That enzyme, angiotensin converting enzyme. And then angiotensin 2 stimulates aldosterone secretion from the renal cortex. So you could have put the renal cortex here, which I guess doesn't make or break this answer, right? But we could have put more detail here. An increase in aldosterone secretion affects sodium reabsorption because it stimulates more sodium-potassium pumps on the basolateral surface, and it also increases the number of open leak sodium and potassium channels on the apical membrane, which increases the number of sodium being actively absorbed into the plasma. So that is correct, right? The 2 things we talked about that aldosterone does, more sodium potassium pumps on the basolateral surface, more leak channels on the apical surface, which facilitates more movement of sodium and also potassium, right? But this was only asking about sodium. The second part of the question, okay, so that was the second part of the question, okay. The second question, where are the cells that secrete renin located? Name 3 stimuli for renin secretion. So these are the granular cells. They're part of the juxtaglomerular apparatus, which is connected to the afferent arteriole. So we could have put that bit here. I wouldn't dock points for that because you did say that they're the granular cells, but you should have put the juxtaglomerular apparatus. That would have been a bit more specific here. The 3 main stimuli are decrease in afferent arterial pressure, renal sympathetic activity, decrease in sodium and chloride concentration in the distal tubular filtrate. So very straightforward. Those are the 3 stimuli that we talked about for renin secretion. And then the third part, how does ANP affect sodium reabsorption? ANP decreases sodium reabsorption in response to increased blood pressure, that's correct, because it closes those sodium channels on the apical membrane, right? So this is really the crux of the answer. It's going to close those leak channels on the apical surface, so sodium cannot get from the tubular fluid into the tubular epithelial cell, and so it cannot be reabsorbed because of that. Okay, it's going to be excreted in the urine. Let's look at some other responses here. This was another great response, pretty succinct. Again, this first part just talking about that RAS system, what converts what into what, be able to explain that. The second part here talks about the juxtaglomerular cells, right? We want to point out that these were the granular cells of the juxtaglomerular apparatus. It's a part of the juxtaglomerular cells, which is located at the afferent arteriole, right? The connection between the distal tubule and the afferent arteriole. And then the stimuli are pretty straightforward here. Decreased afferent arterial pressure, sympathetic activity, and the concentration of sodium and chloride in the distal tubular fluid. And then ANP, the response here, again, very straightforward. We're going to close those leak channels on the apical membrane. That's how ANP decreases sodium reabsorption. Let's see. Okay. If you didn't turn in these review questions, I'll leave this open a little bit. So you have until this weekend, Friday, to put in these extra credit responses. But after this week, I'm going to close these as we move into our exam 4 unit. Okay. Let's look at this third response. So this third response had a few issues. It was kind of okay, but there were some aspects that were incorrect. So this third question, this third response says ANP decreases ADH secretion. This is not true, okay? ADH secretion is not dependent upon ANP secretion. We talked about the stimuli for ADH secretion. That was the plasma osmolarity. So increased plasma osmolarity was one stimulus. The second stimulus was the decreased baroreceptor stretch. So the stretching on those baroreceptors, if that decreased, then we're going to increase ADH. And if plasma osmolarity increased, meaning that you have more concentrated or increased pressure in the plasma, we're also going to release ADH. But then the second part of the question did actually give a correct response. So it decreases water reabsorption. Sodium reabsorption will also decrease. Okay, so no, this was not correct at all, sorry. So this was not correct. Since it decreases water reabsorption, the sodium reabsorption will also decrease as sodium follows water. So this entire response was not correct, okay? That's not how ANP affects sodium reabsorption. The correct response would have been, it blocks the apical sodium leak channels and prevents sodium from moving from the tubular fluid into the cell and therefore into the capillary. Okay, so just be mindful of that. And then the third response here, I also want to point out some issues with this one. The 3 stimuli that were listed here for renin were incorrect, so make sure you're looking over the stimuli correctly. Atrial wall stretching is not a stimulus for renin, neither is ANP secretion. And then plasma volume, this is very vague, right? You're not saying increased plasma volume, decreased plasma volume, you're just saying plasma volume. So be very direct in your response, be very succinct, be clear in what you're trying to communicate so that I can be able to decipher whether it's right or wrong, right? Don't be ambiguous or vague, because then I'm just gonna assume that you don't know the response, right? You don't know a correct response. And then the third response here, this was the one that had some correct components, but also some incorrect components. So the atrial natriuetic peptide affects sodium reabsorption by dilating the afferent arteriole and constricting the efferent arteriole. So while this is correct, this is not how ANP influences sodium. This is how ANP increases GFR. So ANP increases GFR by dilating that afferent arteriole and constricting that efferent arteriole, but the question is only asking us about how ANP influences sodium reabsorption. So this component of the response, was not needed, okay? And then the ending part of the response was correct. This leads to decrease in sodium reabsorption because it closes sodium channels on the apical membrane. So this part was correct. And there was no need to put this first part with the afferent and efferent arteriole in here because it doesn't address the question at all. So make sure you're putting exactly what's needed, exactly what the question is asking, and not additional information just to make a lengthy response. Okay, I hope that was clear and that gave some kind of direction and guidance as to what you should be providing for the essay response. Again, I'm not looking for a lengthy response. I'm not looking for a big, long text. Just be succinct in what the question is asking, be direct, give exactly what's needed, and then make sure it's clear and not ambiguous. If you're talking about a response, say whether it's increased or decreased, and just be very clear about how you're addressing the question. Okay, I don't see any questions pop up, so I'm going to end off here by talking a little bit about the structure of the exam and then some important topics that you want to keep in mind. So first off, I'll make a rubric available. So there will be a rubric in the grading area. It'll tell you exactly how many points are going to be allocated to the content, how many points are going to be allocated to the grammar, how many points are going to be allocated to a clear response, a clear flow in the way you're writing, right? Just don't be putting haphazard sentences together. So there will be some points allocated to each of those components, and that will be clear in the rubric, okay? And the rubric will be available in the assignment. That should be in the grading area. So when you click the assignment, you can right click and it should be able to show you where you can see that rubric. And if you need more guidance on that, I'll probably put a little bit of an instruction in an announcement when I make that go live, just so you can see where to find the rubric. It's going to be available tomorrow at 8 a.m. and it'll be due Friday at midnight. So you'll have a full day, a little bit more than 24 hours to look at the question. You'll be able to go into the question, see the question. You can copy it out into a Word document if you'd like, write your response, paste it back into the text box there. And then when you feel comfortable, you can submit before Friday at midnight. There'll be 50 points total. So remember, this exam is only 50 points, no longer 100. So that'll be 25 points to each chapter. So each chapter will have one question. It'll likely have 3 subparts, very similar to what we had for those checkpoint or those practice questions. So one question, 3 smaller parts to that, worth 25 points each, and one of those for chapter 18, and one for chapter 19. And I will also include 2 extra credit multiple choice questions. So for the benefit of those who want to tackle those multiple choice questions and attempt these extra credit points, you can. If you don't want to, that's okay as well. You will not be penalized for not looking at those questions, but they will be there, again, as extra credit. And then finally, these are some of the main topics that you want to focus on for this exam. So the first 3 of these are from chapter 18. So again, be able to understand and explain the balancing of the Starling's forces, be able to compare and contrast the structure of the proximal convoluted tubule and the distal convoluted tubule in terms of the way the cells look, the function, regulated versus unregulated, the types of tight junction, and so on and so forth. And then obviously the intrinsic and extrinsic mechanisms, be able to differentiate those, okay? For chapter 19, the reabsorption of water. So understand how water is reabsorbed, how that happens in the proximal tubule, how that happens in the loop of Henle, how that happens in the distal tubule and the collecting duct. The fact that it's regulated at the proximal and loop of Henle, and the fact that it is unregulated at the distal tubule, okay? I'm sorry, I flip-flopped that. So it's regulated in the distal tubule and collecting duct, and it's unregulated at the proximal parts. Also, the countercurrent multiplier and the medullary osmotic gradient, the role of urea in all of that, make sure you understand that. The role of these 3 hormones, we kind of talked about these earlier, ADH, absorbing water, aldosterone, absorbing sodium and secreting potassium, and then ANP, decreasing the reabsorption of sodium. And how all of that implies, is implicated in the regulation of blood pressure, right? How blood pressure is regulated with these 3 hormones. And then the stimuli for renin, we talked about 3 stimuli for renin, make sure you know those. We talked about 2 stimuli for ADH, make sure you know those. I went over them earlier. And we talked about 1 stimulus for ANP, which is basically the atrial wall stretch. Make sure you have an understanding of that. Lastly, acid-base balance. So very general, we didn't get into a whole lot of detail with the mechanisms here. So I don't expect you to go into too much detail here, but have a general understanding of why acid-base balance is important, why our cells need to be exposed to a regulated pH in terms of the plasma pH, and why we don't want high pH, such as alkaline environments, or low pH, such as acidic environments, and how the kidneys and lungs kind of regulate that generally, right? So have a good understanding of that generally. Let's see here. Okay, so someone wants to clarify that there's no time limit on when you open and close the exams. That is correct. I will not see when you open the exam, but you can open it, look at the question, you can exit out, you can copy the question somewhere else, you can paste your answer and not submit. So you can paste an answer, go back, edit it, and then when you are ready and comfortable, there's going to be a save and submit button as opposed to just a save button. Okay, another question here. Is the exam timed for our responses once we start on Blackboard? So no, there's no timed component here. The only timed component is the fact that it's due at midnight on Friday. So until midnight on Friday, you have basically all of that time to generate a response, to post it into that text box, and to submit it, okay? Just make sure it's submitted by midnight. Alrighty, let's see if there are any other questions. Okay, so do we answer in bullet type form, like the extra credit examples you showed? That's okay. I'll be okay with bullet points. That'll actually make it a little bit easier to follow. So you can sort of bullet your answers. Again, there will be 3 parts to each question. So let's say I'm asking about the Starling's forces. You can bullet. You can talk about each of those forces and describe what they each do, and then move on to the next, and so on and so forth. So bullets are fine and actually encouraged if you want to use those. Alrighty. I don't see anything else popping up in the chat, so I'm going to take it that that is it in the way of questions. The multiple choice component to that exam will also not be timed. So you again can go in there, look at those questions, and when you're ready to save and submit, that's when that question will save and submit. But you can change your answer, or you could just look at the question, put it off, and select your response when you're ready to submit everything together, okay? And those again will be extra credit, so you will not be penalized if those are wrong or if you don't do those. All right, I think that's all I have for you guys. Definitely reach out if you have questions after I've posted the test. If you have questions about like the logistics of answering the question, or if you have questions about, you know, the question isn't clear or anything like that you want me to clarify, I'm available. And then if you have overall content questions, you can reach out to me between now and tomorrow as well, okay?